Okay, so I'm happy to have an opportunity to tell you about Biosafe. Biosafe is a company that was based on work that was started here 15 years ago when my group and others, collaborators at UNM, started looking at a certain group of materials and looking into their antimicrobial activity and also how they affected Biosafe, as we see it, is really totally dependent on a group of synthetic materials, conjugated polyelectrolytes, and oligomers, smaller versions of the polymers. We use these, we develop their antimicrobial activity. We find they're very active against bacteria, viruses, and fungi. They have some unique properties. There's a bimodal activity. I can't see this because it's, and maybe you can't either, but anyway, they are both light and dark activated. They're strongly fluorescent. That means they emit light that they absorb. We found so far they're environmentally safe. They're degraded to harmless products by sunlight or even by room light. But they have tunable properties for different applications. And specifically, what we've looked at over the last several years, toxicity. We find these are not toxic towards mammalian cells, but they're specifically toxic towards bacteria, viruses, and fungi. They're stable, as I mentioned. We've been looking at primarily as a first product, wipes, and we're talking to various wipe manufacturers, and I think several of them are interested in possibly helping us get through the EPA registration, which is our immediate hurdle at this point. And we've also thought, though, that these materials could be very valuable as coatings, permanent or removable, as solutions, and as mixed formulations. So I'll go on now. I just should mention at the bottom shows a series of fluorescence emitting from our materials for different materials and under different conditions. Now, here are the chemical structures of our materials. We started working actually with the polymers shown on the bottom. These are cationic conjugated polyelectrolytes. We also have looked at anionic ones. And we've also broken these down into smaller units, what we call oligomers, that would be shown by structures 1, 2, and 3. I'll tell you more about the structures later, but continuing, we really think of these as being like a string of beads. And the polymers represent a long string up here. The oligomers are shorter, and but they function in some ways better than the polymers by virtue of their smaller size. And that's been very interesting to discover this. The light-induced antimicrobial activity is designated here, is shown diagrammatically. These materials are, in terms of light-induced reaction, inactive in the dark. But when we irradiate them with light absorbed by the polymer, what we find is we form an excited state, which is highly fluorescent and also highly reactive. And basically the activity in the light-induced antimicrobial activity involves activation of molecular oxygen to an excited singlet state. And the excited singlet state becomes a very strong oxidant and attacks these materials, biomaterials, by probably an electron transfer process and generation of peroxide and other oxidants, what we call in the field reactive oxygen species. Now, how they react can be studied by a number of different techniques. Here I just show with electron microscopy. If we take on the left in the center E. coli bacteria and treat them with one of these materials in the dark, in the light, what we find is that the interior of the bacterium, excuse me, let me go back here. The interior of the bacterium, the contents become cross-linked and fused, and so that clearly inactivates the bacterium. By the same token, when we look at viruses, these are E. coli phages shown here, T4. The irradiation of our materials in the presence of the virus results in destruction of the virus, complete inactivation. We've also looked at yeast cells, and they're inactivated. Now, on the right, on the right here, with the E. coli, we show the dark activation process. And I don't know if you can see it. I can't really control that. But what happens is the cells become empty. Our materials in the dark will attack the cell envelope and rupture it and let the cytoplasmic content spill out. And that's also, we see that the contents are damaged in the dark for the viruses that we looked at up until now. Now, so just to summarize, if we, for example, sprayed a coating onto a surface or wiped it onto the surface, what would happen is after the material dries, it still is on the surface and it's still active against most pathogens. You can tell very easily by putting a black light over the sample and seeing that if it's active, it will glow, as shown here in the center. When it has been used up or destroyed by one means or another, the fluorescence goes away. And so you have a way of telling if your material has coated a surface or not. And as you can see here, you form a very nice uniform coat when you introduce a solution of this polymer or oligomer onto the surface. And you can see that you have a nice active surface for antimicrobial activity. Now, in the last year or so, we've really focused on how fast do these react and how complete is the killing? Are they going to be able to really disinfect the surface or not? And so we've been looking at their reactivity. We did a series of tests on Staph aureus, Staph epidermidis, and Pseudomonas aeruginosa. We use lab strains, but all of these are occur as strongly infecting antimicrobial agents. And what we did first, we've been doing tests around 30 minutes, and we thought that was long enough to give us some reactivity. But here we focused on how complete is the activity, because people don't want even a trace left for these materials. And so here, for example, with Staph aureus, we found that with 30 minutes, we had 6 log kills. That means there's essentially nothing. That means that by doing serial dilutions, we found there was no survival beyond a certain point. Then we decided to try, for example, 10 minutes shorter. And we found even with, for example, with all of these in the light, which is shown by the light bar, and to some extent in the dark, you get really astonishing killing of these bacterial pathogens. For example, Staph aureus, this is 99.999% destroyed. And similarly, we have same with Staph epi, E. coli, not shown here. We have like, again, 5 log kills. And with Pseudomonas, which is the hardest to kill in our experience, with the oligomer shown here, these are all data for this particular oligomer. And you can see for all of them, we have 4 to 6 orders, 6 log kills. And in the dark, we have, in many cases, 4 log kills. And for these 2, not so much. But anyway, they're very reactive.
David, you only have 1 minute left.
Okay. So I'll cut to the quick. We've worked with bacteria and viruses. We use non-envelope viruses in a paper we published in 2011. But this past May, we decided to go after SARS-CoV-2. And we were fortunate to have a coworker available in the North Campus Medical Center, and she had worked with SARS-CoV-2. So we decided to test our materials, and we tested 5 materials against SARS-CoV-2 in the light and in the dark. We found there was no inactivity or of the virus in the dark, but nonetheless, our materials were very, very active in the light. And we used here very nicely to follow a crystal violet stain on another cell that is attacked by the virus. And so you can see wherever the cell is stained, but wherever a virus attacks it, you get a bleaching. And what we found, just to cut it to end up, we found that with our oligomers and with the polymers, all 5 were active, but some were much more active than others. So for example, here, again, we have about 3 logs of inactivity after 5 minutes. Here with another one, after we get 6 log inactivation after 15 minutes. But all of these are active, and we're very excited about this. And we're now pushing our material for EPA approval. And we find that the interesting finding that really comes out of here.
Okay, we're going to have to wrap up, David.
Okay, I'll wrap it up right now.
All right.
The virus cannot be attacked by quats or comparable materials in the dark, but ours function because they are light activated. That's it.
All right, thank you. All right, questions?
Yes, David, this is Ben with Industry Ventures.
Mm-hmm.
I have a question. Based upon your first product being wipes, is there a standard time between wiping a surface down and the usability of that surface?
In our case,
Yes.
yes. They are still active. There's residual activity. And,
Okay, my direct question is, once I wipe a surface, when can I use that surface?
oh, almost immediately. I mean, we'd be using a very volatile solvent. So there'd be quickly a coating. That would happen almost immediately. And of course, the surface would dry in a matter of, depending upon how what the substance was, it would dry probably very shortly. It's acting. If there's a pathogen on the surface while it's wet, there's no question it's continually active. But you should be able to, after I say 5 minutes or so, remove any debris from the pathogen on the surface.
Okay, thank you. That's great.
David, Andrew Moore from Bluestone Venture Partners. Can you elaborate a little bit more about the regulatory pathway, understanding that you need to do submissions to the EPA? You know, what does the timeline of that look like, the cost for going through that, and are there other regulatory agencies you have to work with before you can commercialize this product?
Well, we've been working with EPA. We hired as a consultant a former director of the EPA, who of course is still knows people there quite well. And we've actually negotiated with the EPA a contract where we'll do, we'll provide them with samples, and they'll be testing them not on the regulation tests, but for other use. They're very pleased to find that we have a sample that we have materials that will deactivate the SARS-CoV-2. But the EPA registration, we're hoping we've got 60 published papers in this area. And so we've done toxicity tests, we've done skin tests, mammalian cell tests, and we've looked at the breakdown products. So they can go to our papers. We're hoping they will accept a lot of the data that we've published in peer-reviewed journals. But in any case, we're ready for the test now. And so I hope we can shortcut this. Also, because we have something that's active against SARS-CoV-2, there's an expedited process. And so we hope to do this in, let's say, 3-4 months. And we're virtually ready to send them samples this week.
Got it. And then how long until you can start selling these products and really commercialize this?
That's a good question. That's one reason why we were hoping to partner with a wipes manufacturer. For example, we did some tests a year or so ago with Lysol. And we found that with Lysol wipes, we could add our material and it would make them much more effective at disinfecting the surface. So we think that a partnership of that sort would be a good way for us to proceed.
Got it. Thank you. Just
Any
think
other
about-
questions, or are we ready to wrap up? Okay, go ahead, Andrew. Sorry.
I was just going to say, you know, what does an exit look like for you guys, and when do you expect an exit would occur?
Could you repeat that? I didn't quite.
What does an exit look like for you guys? Who acquires you and when?
We've been talking to people at Benckiser, the parent. They came to us, actually. They were interested in us. And so we've been talking with them. I could see a company like that would be very interested in acquiring our patent portfolio. That's one possibility. We've talked about other wipes manufacturers, for example, or disinfectant manufacturers who are not quite up at the top of the peak at this point, and this might give them a way of getting there. So that's our strategy pretty much at this point.
Got it. Thank you.